Hello, everyone. Welcome to the Bioanalysis 101 webinar series. In this video, you will be able to learn about the immunogenicity and anti-drug antibodies and use the knowledge for your job search or apply in your regular day-to-day job in bioanalytical department. In this video, I'll be going over the overview of immunogenicity. What is immunogenicity? What are anti-drug antibodies? What is the impact of anti-drug antibodies on safety and efficacy? And the overview of regulatory recommendations for immunogenicity assessment. First of all, what is biologics and gene therapy product? Biologics and gene therapy products are a class of molecule that include antibody therapeutics such as Humira or adalimumab, the recombinant proteins and enzyme replacement therapies such as Fabrazyme, and gene therapies such as Zolgensma. These biologics and gene therapy products are administered to the human for the correction of certain diseases, but these are inherently immunogenic, which means they have their properties to evoke the immune response. Once the drug or these biologics and gene therapies are manufactured through a biological process, which includes the cell culture and purification, those drugs are known as formulated drug substances. And these drug substances are administered to the host, and that is human, that would provide the drug effect. The drug effect can be divided into two groups. One is intended effect, where it's a treatment benefit. As for example, if someone is suffering from cancer and the anti-cancer drug would reduce the cancer. But there are other unintended effect as well. One of them is generation of immune response. And overall, the immune response can be divided into two types. One is humoral immune response, that is antibody response, and the other one is cell-mediated immune response. And the incidence, that is the magnitude and the severity of these immune response depend on various factors. One, it could be the nature of therapeutic product. The manufacturing process and impurities also play a vital role in the generation of immune response. The host genetic factor itself has an important role to play in the generation of this unintended immune response. And the site of administration, such as intravenous, intramuscular, or parenteral, has its own effect. So what is anti-drug antibodies? Anti-drug antibodies are generated in response to protein and gene therapy products, and it is a part of humoral response. These anti-drug antibodies are specific to the protein or gene therapy products. And these anti-drug antibodies bind to the therapeutic product, and these ADA binding to the drug may be just binding, or it also neutralizes its effect. And ADA, these ADA, we also call ADA for anti-drug antibodies, may impact safety and efficacy. So how does anti-drug antibody impact safety and efficacy? First mechanism is ADA binding and sequestration of drug may result in decreased bioavailability. As for example, in this figure, if the person or the clinical subject does not have any antibodies, they have a higher availability of drug that could bind to the target, that could bind and enter the target tissues. But if there is higher concentration of ADA, that could bind and decrease the bioavailability and therefore exposure of the drug. ADA may also promote formation of anti-drug antibody complex with the drug and increase its clearance. In this figure, as you can see, this upper one is without ADA or anti-drug antibody, and the lower one is with anti-drug antibodies. X-axis represents the time, Y-axis represents the concentration of drug. And as you can see, this is a representative figure where the presence of ADA rapidly decreases the concentration of drug at second time point compared to the presence of ADA. The third mechanism by which it can affect the safety and efficacy safety is that there may also be a subset of total antibody that can mask the active site of the drug and inhibit its activity. And these are called neutralizing antibody. This neutralizing antibody masks the active site and therefore inhibits the function of the drug. And therefore they hinder the intended effect of the drug. The anti-drug antibody may also impact the safety by inducing anaphylactic reactions, cytokine release syndrome, complement-mediated lysis, and antibody-dependent cell cytotoxicity. Therefore, FDA recommends adoption of a risk-based method to evaluate and mitigate immune response. And also, FDA also states that any given approach to assessing and mitigating immunogenicity is determined on case-by-case basis and depending on the risks. If there is a higher risk, you need to pay more attention to the immunogenicity mechanism of the anti-drug antibody generation and its effect compared to the molecule that has a low risk of generating immune response. And some of the immunogenicity assessment includes detection of total anti-drug antibodies, detection of neutralizing antibodies, characterization of isotypes, detection of IgE, where there is higher incidence of anaphylaxis, detection of cross-reactive antibodies, if the molecule or the drug you're working with is homologous to endogenous counterparts, and characterization of antigenic epitopes if it's a multi-domain protein therapeutics. In summary, I would like to conclude with the following take-home message. Biologics and gene therapy are inherently immunogenic because of its nature, and it can generate unwanted immune response, which is also known as immunogenicity. The immunogenicity includes anti-drug antibodies and T-cell response, and anti-drug antibodies are a part of humoral immunity. These anti-drug antibodies may impact safety and efficacy. The regulatory agency requires sponsors to monitor immunogenicity as a part of their clinical trial. With that, I would like to stop the video here and would like to thank you for listening to this. Please like and subscribe, and to get more more videos on the field of bioanalysis. I'll be including more of these kinds of videos for such as assay development, bioanalysis, and other relevant videos. Thank you.